Welcome to the fascinating world of genetics and heredity. In this video, we'll explore how traits are passed from one generation to the next, shaping everything from our eye color to our risk for certain diseases. Genetics is the study of genes, the basic units of heredity, and how they influence who we are. Heredity refers to the process by which parents pass down physical and biological characteristics to their offspring. Understanding these concepts not only helps us appreciate our uniqueness, but also reveals the connections we share with our families and ancestors. Let's begin our journey into the building blocks of life. Genes are segments of DNA that act as instructions for making proteins, which perform most life functions. Each gene carries specific information that determines traits such as hair color or blood type. Humans have thousands of genes, and each one plays a unique role in our development and health. Genes are inherited from our parents, with half coming from our mother and half from our father. This combination creates the diversity we see in every individual. Understanding genes is the first step in unraveling the mysteries of heredity. DNA, or deoxyribonucleic acid, is the molecule that contains the genetic instructions for life. Its famous double helix structure stores all the information needed to build and maintain an organism. DNA is found in nearly every cell of our bodies, tightly packed into structures called chromosomes. These chromosomes ensure that genetic information is accurately copied and passed on during cell division. The sequence of DNA bases forms the code that determines our unique characteristics. Without DNA, life as we know it would not exist. Chromosomes are thread-like structures located in the nucleus of each cell. Humans typically have 46 chromosomes arranged in 23 pairs. Each parent contributes one chromosome to each pair, ensuring a mix of genetic material. Chromosomes carry our genes and play a crucial role in heredity. Any changes or errors in chromosomes can lead to genetic disorders. Understanding chromosomes helps us see how genetic information is organized and transmitted from one generation to the next. Gregor Mendel, an Austrian monk, is known as the father of genetics. In the 1800s, Mendel conducted experiments with pea plants, carefully observing how traits like flower color and seed shape were inherited. His work revealed that traits are passed down in predictable patterns, laying the foundation for modern genetics. Mendel's discoveries introduced the concepts of dominant and recessive traits, which are still fundamental to our understanding of heredity today. Traits can be dominant or recessive. A dominant trait will appear if at least one parent passes down the gene for it, while a recessive trait only appears if both parents contribute the gene. For example, brown eyes are often dominant over blue eyes. This explains why some traits seem to skip generations or appear unexpectedly. Understanding dominance and recessiveness helps us predict the likelihood of inheriting certain characteristics. Punnett squares are simple diagrams used to predict the possible genetic outcomes of a cross between two parents. By organizing the parents' genes in a grid, we can see the probability of their offspring inheriting specific traits. This tool is widely used in classrooms and laboratories to teach and study heredity. Punnett squares make it easier to visualize how dominant and recessive genes interact. Genotype refers to the genetic makeup of an organism, the specific combination of genes it carries. Phenotype is the observable physical or biochemical characteristics, such as height or blood type, resulting from the genotype and environment. Two individuals can have the same phenotype but different genotypes. This distinction is key to understanding how genes and environment work together to shape who we are. Alleles are different versions of the same gene. Each person inherits two alleles for each gene, one from each parent. These alleles can be identical or different, leading to variations in traits. For example, the gene for eye color has several alleles, resulting in blue, brown, or green eyes. The combination of alleles determines the traits we express. If both alleles for a gene are the same, the individual is homozygous for that trait. If the alleles are different, the individual is heterozygous. This distinction affects how traits are expressed. For example, a person with two dominant alleles for brown eyes is homozygous, while someone with one dominant and one recessive allele is heterozygous. These terms help geneticists predict inheritance patterns. Mendel's laws describe how traits are inherited. The law of segregation states that each parent passes only one allele for each gene to their offspring. The law of independent assortment explains that genes for different traits are inherited independently of each other. These principles form the backbone of classical genetics and help explain the diversity seen in living organisms. Mutations are changes in the DNA sequence. They can occur naturally or be caused by environmental factors like radiation. Some mutations have no effect, while others can lead to genetic disorders or beneficial adaptations. Mutations are a driving force behind evolution, introducing new traits into populations. Understanding mutations helps us grasp how genetic diversity arises. Mutations or errors in genes can cause genetic disorders. Examples include cystic fibrosis, sickle cell anemia, and Down syndrome. These conditions can be inherited or occur spontaneously. Genetic testing and counseling help families understand their risks and make informed decisions. Advances in genetics are leading to better treatments and hope for those affected. X chromosomes determine an individual's biological sex. Humans have two sex chromosomes. X and Y. Females typically have two X chromosomes, while males have one X and one Y. The combination of these chromosomes influences not only gender, but also the inheritance of certain traits and disorders. Understanding sex chromosomes is key to studying patterns like X-linked inheritance. Some genes are located on the X chromosome, leading to X-linked inheritance patterns. Since males have only one X chromosome, they are more likely to express X-linked traits such as color blindness or hemophilia. Females with two X chromosomes are often carriers. This explains why certain conditions are more common in one gender than the other. Pedigree charts are diagrams that track the inheritance of traits through generations of a family. They use symbols to represent individuals and relationships, making it easier to identify patterns of inheritance. Pedigree charts are valuable tools for geneticists and doctors, helping them diagnose genetic conditions and predict risks for future generations. For a cell divides, it must copy its DNA so that each new cell receives a complete set of genetic instructions. This process, called DNA replication, is highly accurate but not perfect. Sometimes errors occur, leading to mutations. DNA replication is essential for growth, development, and the maintenance of life. Genes provide the instructions for making proteins, which are crucial for nearly every function in our bodies. The process of protein synthesis involves two main steps: transcription and translation. During transcription, DNA is copied into RNA. In translation, the RNA is used to build proteins. This process ensures that our cells function properly and respond to changes in the environment. Genetic variation refers to the differences in DNA sequences among individuals. These differences are the result of mutations, recombination, and other processes. Variation is essential for evolution, as it provides the raw material for natural selection. It also explains why no two people except identical twins are exactly alike. Genetic engineering is the process of altering an organism's DNA to achieve desired traits. Scientists use techniques like gene editing to add, remove, or modify genes. This technology has led to advances in medicine, agriculture, and industry. While genetic engineering holds great promise, it also raises ethical questions about how far we should go in modifying life. Cloning involves creating a genetically identical copy of an organism. This can occur naturally, as in identical twins, or be done artificially in the lab. Cloning has been used to reproduce plants, animals, and even cells for research. While it offers scientific benefits, cloning also sparks debates about ethics and the value of individuality. Genomics is the study of all the genes in an organism, known as its genome. Advances in genomics have made it possible to sequence entire genomes quickly and affordably. This field is revolutionizing medicine, agriculture, and our understanding of evolution. Genomics helps identify disease risks, develop new treatments, and trace our evolutionary history. Epigenetics explores how environmental factors can influence gene expression without changing the DNA sequence. Factors like diet, stress, and exposure to toxins can turn genes on or off, affecting health and development. Epigenetics shows that our lifestyle choices can impact not only us but also future generations. Human Genome Project was an international effort to map all the genes in human DNA. Completed in 2003, it identified over 20,000 genes and unlocked new possibilities for medicine and research. This project has paved the way for personalized medicine, where treatments can be tailored to an individual's genetic makeup. Genetic testing analyzes DNA to identify changes or mutations that may cause disease. It can help diagnose conditions, predict risks, and guide treatment decisions. Genetic testing is becoming more accessible, empowering people to make informed choices about their health and family planning. Gene therapy is an experimental technique that uses genes to treat or prevent disease. Scientists can replace faulty genes, introduce new ones, or turn off harmful genes. While still in development, gene therapy offers hope for treating genetic disorders that were once considered incurable. Not all traits are inherited in the same way. Some follow simple Mendelian patterns, while others are influenced by multiple genes or environmental factors. Examples include autosomal dominant, autosomal recessive, and polygenic inheritance. Recognizing these patterns helps geneticists predict how traits and diseases are passed down. Some traits like height and skin color are controlled by multiple genes. These are called polygenic traits. The combined effect of several genes leads to a wide range of possible outcomes, explaining the diversity seen in complex traits. Polygenic inheritance makes predicting these traits more challenging. Genes are not the only factor shaping who we are. The environment, everything from nutrition to experiences, also plays a significant role. For example, identical twins raised in different environments can develop different traits. This interaction between genes and environment is key to understanding human diversity. Genetic counseling provides information and support to individuals and families at risk for genetic disorders. Counselors help interpret genetic test results, discuss risks, and guide decision-making. This service is especially important for those planning families or facing inherited health conditions. Genetic drift is a random change in gene frequencies within a population. Unlike natural selection, which is driven by survival advantages, genetic drift occurs by chance. It can have a significant impact on small populations, leading to the loss or fixation of certain traits over time. Natural selection is the process by which traits that improve survival and reproduction become more common in a population. Over generations, beneficial traits are passed down while less advantageous ones fade away. Natural selection, along with genetic drift and mutation, drives evolution and shapes the diversity of life. Gene flow occurs when individuals from different populations interbreed, exchanging genetic material. This process increases genetic diversity and can introduce new traits into a population. Gene flow is important for the health and adaptability of species, especially in changing environments. Selective breeding is the process of choosing parents with desirable traits to produce offspring with those traits. Humans have used selective breeding for thousands of years to improve crops and domesticate animals. While effective, it can also reduce genetic diversity and increase the risk of inherited diseases. Genetic markers are specific DNA sequences used to identify individuals or track inheritance. They are valuable tools in research, forensics, and medicine. Genetic markers help scientists map genes, study populations, and solve crimes by matching DNA samples. Mitochondrial DNA is found in the mitochondria, the energy-producing structures in cells. Unlike nuclear DNA, it is inherited only from the mother. Mitochondrial DNA is used to trace maternal ancestry and study evolutionary relationships among species. Genetic fingerprinting is a technique used to identify individuals based on their unique DNA patterns. It has revolutionized forensic science, making it possible to solve crimes, identify remains, and establish paternity. Genetic fingerprinting relies on analyzing specific regions of DNA that vary greatly among people. The rapid progress in genetics raises important ethical questions. Issues include privacy, consent, genetic discrimination, and the use of genetic information. Society must balance the benefits of genetic research with respect for individual rights and the potential for misuse. Personalized medicine uses genetic information to tailor treatments to individual patients. By understanding a person's genetic makeup, doctors can choose the most effective therapies and avoid harmful side effects. This approach is transforming healthcare and improving outcomes for many diseases. Families facing inherited conditions often seek genetic counseling for guidance. Counselors explain the risks, inheritance patterns, and options available. This support helps families make informed choices about health, reproduction, and managing genetic disorders. The future of genetics holds incredible promise. Advances in technology are making it possible to edit genes, cure diseases, and even prevent inherited conditions before birth. As our understanding grows, genetics will continue to transform medicine, agriculture, and our view of life itself. Genetics isn't just for scientists, it affects our daily lives. From ancestry tests to personalized diets, genetic information is becoming more accessible. Understanding genetics empowers us to make better health choices and appreciate the diversity of the human experience. Genetics is the foundation of evolution. Changes in genes over time lead to the development of new species and the adaptation of organisms to their environments. Studying genetics helps us understand the history of life on Earth and the processes that drive biodiversity. Genetics plays a vital role in agriculture. By understanding and manipulating genes, scientists can create crops that are more nutritious, resistant to pests, and adaptable to changing climates. Genetic advances are helping to feed a growing global population and ensure food security. We've covered the basics of genetics and heredity, from the structure of DNA to the impact of genes on our lives. Genetics connects us to our families, shapes our health, and drives the diversity of life. As science advances, our understanding of genetics will continue to grow, offering new opportunities and challenges. Thank you for joining this journey into the wonders of heredity.